Hello and welcome to our first virtual Darwin Day at Bridgewater State University. These series of lectures that are available to you are in celebration of Charles Darwin and the theory of natural selection. My name is Maria Armour. I am a staff member and instructor at Bridgewater State University in the Department of Biological Sciences. I teach anatomy and physiology, and one of my specialties, and I'm a specialist in bat biology. So I'm going to be talking a little bit about bats in this presentation. But what I'm here to discuss about today is looking at morphology. Morphology, if some of you don't know, this is the study of the structure of the body, whether it's our hands in humans or a bat's wing, and looking at that structure and seeing how it allows that organism to evolve or make a living. So the topic or the title of my talk today is, Does morphology have a place in today's evolutionary biology? So a lot of us have Instagram accounts, right? And I personally, in my Instagram account, I have a ton of animal pictures, and some of them are cute squirrels and, making a fool of themselves at the bird feeder. Others are from natural history museums across the world. A lot of them are nature photographers. And what I love is, as you scroll through these, your Instagram account, you get to see all these unique shapes of the organism. And what I want you to start to do is, if you do have an account like this or, TikTok, start scrolling through and look for certain accounts that are based on evolution and based on animal or animal behavior. A few of the pictures that I came across, we have some beautiful, gorgeous pictures that I've enjoyed looking at. Many from Joel Sartore, who's a Natural Geographic photographer. The Center for Biodiversity. This one I like. It's the Pika, and as you can see, this is a non-scientific anatomy right here. The megaphone and heart sweater. But as you're flipping through, start asking questions of these images. And what I'm talking about in that is applying the term morphology to your everyday life. If we're looking at, you're patting your cat or something while you're at home watching TV, start looking at the anatomy of that cat, the structures or morphology, and say, why does it have the whiskers a certain direction? Or why is the tongue, when they lick you, why is the tongue really rough? What allows that organism to interact, to feed, or so you're going to kind of apply the functionality onto the morphology. So really defining what morphology is looking at it's a descriptive science, and it's looking at the structures of the body and analyzing that form, an organism's form. How do the structure of something allow that organism to perform a certain function? And what the form of something is due to its phenotype. So our genes are the genotype, our DNA, and they dictate what phenotype, the physical aspect of our form is going to be. So understanding how organisms function and how diverse phenotypes have evolved is really what the core study of morphology is. And what it can do is it can shed a lot of light on the mechanisms that allow changes to occur during evolution. Remember, genotypes, when there's a change in our genotypes in our genes, it does dictate what phenotype is being expressed. So you can, the early scientists have looked at morphology and said, well, you have a five-fingered upper limb, and this organism has 5 fingers. You must be similar, evolutionarily speaking. So you must have a common ancestor. Well, let's take another look at those images, right? So if we take a closer look at a couple of those images that we scroll through in Instagram, start to ask questions. So the viperfish. Well, this viperfish, it has these nice huge fangs, this large eye. It is kind of muted in color. This is a deep sea fish, so it feeds in the deeper water where there is no light source. And its fangs and its large eye allow it to find its prey. We then have over here, this is a bird of paradise, and a huge, beautiful plumage on this male. And what this plumage allows is it allows the male to attract females, beautiful colors. And then in the center, we have this very interesting looking mammal called an aye. It is a young aye in this picture. A couple of things I'm noting is its long digits, its long fingers. And if looking into the biology and ecology of this organism, the aye has these extremely long, skinny fingers. And what it allows it to do is to go into termite nests, and actually it puts its finger in there and takes it out kind of like you would see a stick, and filter out, get all those termites and munch on them. Another cool thing are these beautiful large ears that are downward shaped. They kind of couple downward. This allows the aye to actually pick up sounds that are being made by the termites. So how can morphology be applied to general today's biology? Is this a science that is being applied today? It is. There is tons of research being conducted right now, especially in the field of comparative morphology, where you're comparing structures from similar species and seeing how the different structures are allow those organisms to interact with each other, what the function behind it, so functional morphology, function behind a certain structure. So there's a lot that can be learned as a functional morphologist, as a morphologist, and can be applied to today's sciences. So it wouldn't be a Darwin Day talk without actually talking about Charles Darwin. He was one of the early morphologists. He was a naturalist, and during his days on the Beagle, when he went around to where the Galapagos Islands are, he started thinking as a morphologist, as a naturalist. And so Darwin's study on the Galapagos finches has been used to help understand, and it actually helped him establish the theory of natural selection. Morphology is a way for us to understand evolutionary processes, and it's been applied from early naturalists, even far prior to Charles Darwin times. So it's historically been used as the model for studying speciation and adaptive radiation. It also helps explain the diversification of similarly related organisms that exploit unique niches like our Galapagos finches. Now, the field of morphology was an original way scientists connected evolutionary patterns and processes among similar groups of organisms. Evolutionary trees were constructed on shared derived characters. So these are, we'll see a bunch of trees in today's lecture. They're going to, some of them are going to be using morphological characters to connect and see if there is a evolutionary connection, a common ancestor. But this is how morphology has kind of evolved over the years, how it's been applied in evolution greatly in early years. And right now, since genetics and molecular biology has come along, there's a little, it's not as heavily used in evolution, but we'll try and understand if it still has a place. Now, I love finches. I love birds. I appreciate beautiful things, especially when it's biological. But as I said earlier, I am obsessed, passionate about one group, one large order of mammal, and that order is the bats. There we go. Okay, so my study, my taxa of choice is going to be Chiroptera, which are our bats. Huge diversity in bats. So bats make up the second largest order of mammal. They are not flying rodents. They are not flying mice. They're actually closer, evolutionarily speaking, closer to primates and to whales than rodents are, than they are to rodents. So what bats do is, as a biologist and an evolutionary thinker, I love to use bats as my species of choice and use it as an example to teach basic concepts in evolutionary biology. The reason being is that they have an extremely diverse extremely diverse families, and within those families, their genera and species are extremely diverse as well. Not only diverse in how they look, and we can see a couple of them right here. This one's one of my favorite. It's the wrinkled face bat. But we have, and this is like a pom-pom bat, it's called Morphology. This is a vampire bat, and so on. So they have these diverse structures on their faces, and their body is different. So their morphology is very, very different. But if you really look at their morphology, it's actually dictating or has a direct correlation with what food they eat, how they get around. Are they echolocators? Some are not. The ones that echolocate have smaller eyes, larger ears. The ones that do not echolocate, like this beautiful one, this is in a family that is only uses sight to get around. So we can tell a lot by its structure. This is a type of bat that is, it has this beautiful plumage, only the males have it, and this is connected to scent glands. So this large plumage, when it becomes erect, it actually produces a scent that attracts the female. So very, very diverse. And I love using bats because they're so diverse. Now there's 1,400 plus species. And when I started out in bat biology, 15 years ago, there was only 932, I think. And because in the last 15 years, because of genetics, molecular studies, and phylogenetic studies, we have seen a huge increase in our speciation to properly identify and tease out these very, very diverse group of mammals. So as I mentioned, I've studied bats for about 15 years now. My early career in bat biology was spent, my schooling was spent at the American Museum of Natural History, where I happily sat in a very cold lab with a bunch of dead bats, rarely saw people, and it was a fantastic experience. What I did during my my schooling and for my thesis, what I did was I looked at how bats, their morphology, how bats folded their wings into their body. And I looked down to the detail of the gross anatomy. So as you can see here, this is just a couple of ways bats fold their wing. They can fold it like us, that we have a, flexion and extension, right? Some bats fold like an accordion. And some bats only have one folding, so this is their digits. And I use this to see if there was any correlation between the morphology of folding, the action of folding, and their evolution. So the evolution and speciation of this very diverse order. I applied these morphological characteristics and I put them on a tree. the bat tree, and that's what you see here. This is it's no longer accurate because so much has changed over the years, but this is my phylogenetic tree that I use to, that I applied certain characteristics in my study to. A little shout out to bats. I know they're getting a poor record right now. It's not their fault. They are a very gregarious and very beneficial group of organisms. Now, as much as I love being a primate, I would love the ability to fly, and I am always very confused of why humans never evolved wings, right? Bats did. Now, bats are the only only order of mammal that actually has evolved the ability to perform true powered flight. And by that, I mean it has its wings, its fingers have elongated through evolution. It has retained the webbing, so a membrane in between its fingers other than the first digit. And that allows it to, you can see the billowing effect of its wing membrane. Also has evolved these beautiful muscles both on the anterior and posterior, so front and back of the bat. Now, I don't have those capabilities. That was due to something called developmental constraint. So during as humans in our embryonic development stages, we are limited to the sort of phenotypes that can evolve. So like wings, we're limited to certain things that have basically, we will not be evolving wings like that of the bat. Now, the development of wings or some advantageous feature may actually be fatal and interrupt other aspects of an organism's development. Or perhaps these features would require so many other drastic changes during developmental stages that they are unlikely to arise through a random mutation, through evolution. So I just have to accept the fact that I am not going to evolve wings. My offspring and future offspring will very unlikely develop wing patterns. Now, all vertebrates evolved from some common ancestor. We share homologous structures on our limbs that have diverged in their functionality. So all vertebrates come from a common ancestor. These homologous structures, for instance, if we look at the forelimb of these organisms, a human, lizard, bat, and whale, we know that these organisms are performing different functions, but we do have a common ancestor there. So if we look at these vertebrates, we see that the forelimbs that are homologous are based on their bone division. They have a stylopod, which is the humerus, or femur. So this is forelimbs and hind limbs. So it's a 1, 2, 3. Then we have the zygopod made up of the radius and ulna and the tibia and fibula for the lower limb. And the autopod, this is the carpals, our wrist joints, the metacarpals, and phalanges, our fingers. And for the lower limbs, tarsals, metatarsals, phalanges. So this is where vertebrates have homologous structures, meaning that they have some relationship with a common ancestor. But because the different arrangements of these bones within each lineage, the forelimbs and hindlimbs are able to perform different functions, like flight, or in the case of a whale, like a paddle. Here we see how the structure, the same structure that has a common link to it, performs very, very different functions. So how do we even know that we're related? How can we identify that? Well, we use something called a phylogenetic tree. You may have heard it as an evolutionary tree, one and the same. We base this on, so we're these organisms that we want to see if they're linked, we base it on shared derived characters. Characters like hair in mammals, or in mammary glands, or nails, fingers, digits, something that you can have hundreds upon hundreds of different characters that you will score, and then that scoring will be placed onto a phylogenetic tree. Now it's organizing these trees organize biological diversity. They also show a pattern of evolutionary descent from a common ancestor. And they show speciation events where you have a common ancestor, and if a speciation event occurred, you'll see a branch. So that's the branch of a tree. The types of trees we see, and you may see here or elsewhere, we have different shapes, so you can show it differently. We have this classic tree branch here. We have one that is horizontal, so you have a speciation event happening whenever you see a branch. Common ancestor is always, the past, the ancestor is back here, and the descendants are at the tips of the branches. And then in the instance of the tree of life, when there is a lot of speciation events occurring to fill in one area, then you usually see it in a circular shape. So what do these branches mean? So as I said, you have a common ancestor. So this common ancestor is a common ancestor of descendants 1 through 4, right? Descendants are at the tips of the tree. Every time you see a branch, this is a speciation event. That means that one species divided into 2. Then we come up to another possible common ancestor. The common ancestor that would be here is the common ancestor of descendants 2, 3, and 4. This, but not 1. And then we see another one. This is the common ancestor where it would be for organisms 3 and 4, but not 2 and 1. So we can use these trees, and you can date the trees depending on, especially when you involve fossil record. So you can date the trees, and one of the take-homes for a phylogenetic tree is the more data, the more characters you apply, the stronger your results are. So the stronger the evidence is for that particular speciation event or hypothesized connection. Darwin was one of the first to make this connection of a phylogenetic tree. Of course, it wasn't called that back then, but what he did is he used phylogeny, he used this tree branching effect in several of his papers and books. One was on the primate relationships, hypothesized primate relationships. And what a phylogenetic tree is it is a prediction, a hypothesized connection based on the evidence that has been gathered. Here we have these Darwin trees. This is one of his early ones, his most famous one from 1837, where he wrote on it, and I love this. I think, you as a scientist, you are skeptical. You want to apply and make statements based on evidence, and you have to be critical of both of yourself and your structure of the scientific method. And Charles Darwin did just that. So we see the evolution of these trees going all the way to, this is the tree of life right here. And this is one of those circular trees, as I said. And you can see if all the speciation event happens, this is covering all of life. So bacteria and birds and mammals and all of those, fungi, all of those wonderful things are fit into this tree of life. So as I said, phylogenetic trees are going to show a hypothesized evolutionary relationship among the group of organisms that you're studying. So I could do a phylogenetic tree on all the organisms in the world that have been known. So that's a very large tree. That's the tree of life. Or I could take my subset, my taxa of interest, bats, and I could make a phylogenetic tree just based on those 1,400 species. And what it does is it's going to depict evolutionary descent from different organisms and identify a common ancestor. So again, we have our common ancestors every time we see a speciation event. At the ends of the trees, those are the descendants. Some descendants, so in certain formats of a tree, you can see that the descendants are still in existence. Some are fossilized, so they are no longer, they're extinct. What we use for phylogenetic trees is we use a data set. And that is, in the old days of natural history and evolution, they were mainly morphologically based. Right now, with the onset of genetics and molecular studies being cheaper and more available, we see molecular is also involved. And this is, there has been a great transition into molecular studies when applying it to a phylogenetic tree. Sometimes we see behavior, but that's rarely used. The principle of parsimony, this is just something that when we run our data set, so it's a computer programs out there that run it. When we run our data set, we're going to produce a lot of options for potential hypothesized relationships. The principle of parsimony states that it is usually the simplest explanation that can explain the data is preferred. So more simple, the less gains or losses of something, or the less mutations, if we're talking about molecular, the more likely it would happen. So let's apply this knowledge on phylogenetic trees using my wonderful example of bats. To make a phylogenetic tree, I'm going to identify characters. We're going to do morphology. These are something that is observable. So my characters are up here, the hand, the eyes, the leg membrane, body hair, and ears. Within that character, you have different states. So states go from starting at 0, which is the most ancestral state, and then they can go up to 3 or 4. And as you increase in number of state, it's further and further away from the ancestral condition. So our hand, for instance, 0 would be that the fingers are separated. 1 would be that the hand is long and has a membrane. Now, with this, we have our group. This is the taxa that I'm going to use. This is considered the in group. And I'm going to try and see how are these 6 bats related to one another. And I'm going to score their state for each character. Now, in order to really do this, what I need is a outgroup. So I said this was the in group. Well, I need something to another species to compare it against. And what this does is using an outgroup is going to root a tree. It's going to tell me what the common ancestor is. So this is a common practice that is used. So who am I going to use as an outgroup? Well, I would like to use myself. Right? Okay, so I'm going to use myself as the outgroup that's in this instance closely related to, or hopefully closely related to, our bats. So now I scored my phylogenetic tree, I scored my character matrix. I looked at the characters, hand, eyes, legs, and hair, and I scored it for each of my species, including myself, right? And what I came out with this was my phylogenetic tree. So I have my common ancestor back here. I have my outgroup, which is myself. and then my six bat species. And what we see is every time we have a branch, that is a speciation event. It's a separation. And as we see these nicks, these are either gains or losses. So a gain of wings occurred here. So this is the common ancestor that had wings that include all of my species. Whereas big ears were gained about here to these two common ancestors. White hair was here. And so what we're seeing is that we can actually relate historically the where our speciation events and these characters actually fall on a tree. So with bats, early on, if we looked at only or applied only the morphological tree, morphological characters, this is the phylogenetic tree that we came up with. And what we saw, this has been a long-standing phylogenetic tree for bats, but again, it's only using morphology. And based on the morphology, we saw that this large group, the megabats called Pteropodidae, were actually very distant than all other species of bat here, so all other 18 families. And that is due to the evolution of flight and echolocation. Now, there is warnings to using only morphological characters because it will lead to homoplasy. This is when we have shared similarities in phenotype, in the structures, that is not due to a common ancestor, but may be the result of evolution or a reversal, a loss of certain structures. So this tree, although it's nice having the megabats up here and the microbats, this is incorrect, even though it's been long-standing that this is the phylogenetic tree. We had molecular data come up in the 80s and 90s and throughout the 2000s, and it is changing the way we're looking at phylogeny. So molecular data is going to highlight the prevalence of convergent evolution in phenotypic traits. So convergent evolution, it has the same function, but the structure is not linked to a common ancestor. So molecular data, how is that used? Remember, if you guys know by now what DNA is, this is the genetic code. The genome has been sequenced for many organisms now. Now, DNA contains the instructions for building an organism. And those genes, every single nucleotide position in the DNA, the adenine, guanine, cytosine, thymine, each position in a long strand of DNA is a character in itself. And there are 4 states, so we're talking phylogenetics, there are 4 states for each character. So depending on what nucleotide position is, if there's an A there, if there's an adenine, that's going to be one state. If there's a guanine, that's going to be another state. And so what looking at the DNA does is it allows us to really look at and acquire an enormous amount of genetic data and characters to apply to a tree, making that tree stronger, the evidence stronger. Every time there's a mutation in an organism's DNA, that means that there is a variation in its genetics. So applying molecular data into phylogeny is going to create much stronger trees. And this is really why I'm just telling you. So this is, let's say these are 5 different species, and these are the DNA strands. And based on where that mutation is, or where the nucleotide position is, each one of these nucleotide positions is a character in itself. And if we look at it, we can actually see, so these two are very similar species other than one nucleotide, one state. So this has a cytosine, this has a guanine. So these 2, 3, and 4 species are closely related to one another. whereas 2 and 5 are also closely related to another, but they because they only have 2 nucleotides difference. So if I put this on a tree, 2 and 5 are going to be close together, and 3 and 4 are closer together. Now 1 is going to be considered an outgroup for me because it is the furthest away. But we can see where these speciation events have occurred. So now let's take another fresh look at our bat tree. When we applied all of the genetic data that we had over just in the last 5 or 10 years, we've seen the mega and microchiroptera division, those suborders, flip on its head. And now we're seeing that our megabats, which would be up in this suborder called Enoterochiroptera, and these are some microbats that have can echolocate. These are grouped, they're more similarly related, and they have a common ancestor. And this group, the Yangochiroptera, this is all microbats, but what we're seeing is that the speciation events and timing is totally different than what we're used, what we established based solely on molecular data. So you don't have to memorize this or anything, but what I'm showing you is that there is a big change, and they took this phylogenetic tree of bats and turned it on its head. So what we thought of bats and their relationship to one another, these different species, has changed drastically over the years. One cool thing that you can do when you're looking at these characters with different states is you can actually apply them and map them onto a already established phylogenetic tree. So looking at these different color-coded, it coincides with different echolocation types. It coincides, each one of these is a different state for the character of echolocation production, so what type of sound they're producing. And so we can actually follow and identify if a particular character state, particular morphological or molecular structure is linked to a common ancestor, or we can tell if it's a convergent evolution happening. So if it's not, so these in yellow would be examples of convergent evolution because we cannot find a common ancestor to link the two. Now, using morphological data in phylogenetic studies is still vital, it's still important, and that is because of fossil evidence. Now, fossils, we cannot extract DNA from, so we don't have that capabilities of actually looking at the genetics of fossils. But what fossils do is they allow us to do a time scale on when certain characters evolved, and that is based solely in morphology. So when did bat wings evolve? Based on the structures of the ear and the larynx, when did echolocation evolve? All of that information we know because of fossils. And so we can time date, and fossils allow us, by using morphological information, we can date our trees, and that is applying the molecular clock. This is a way that we can measure divergence that has occurred in evolution. So our current phylogenetic trees have a combination of characters. The majority, about 98%, is molecular now, it's genetic. Only less than 2% is morphological, and that is stems from the fossils, needing to incorporate fossils into our study. What we're seeing is a great decrease in morphological study and increase in molecular studies. So my question to you, is morphology still pertinent in current day phylogenetic studies? It's still used. It is important, especially when we're talking about fossils, because we can timescale our divergence and these evolution of certain structures. So it is still important. Modern day morphology and how it's being applied outside of phylogenetic studies, it is has become gone from the naturalist way of sketching and drawing out your different structures, and it's gone and increased in its capabilities and gone into computerized 3D imagery, looking at things like morphometrics, applying it in biomechanical modeling. There are a few examples that we have. The modeling, for instance, out in at Brown University, they created a robo-bat, so a bat robot based on the wing structure. So they apply, this is a biomimicry, so they are applying a certain structure onto a robotic, kind of mimicking the biologic into a robotic structure. So if you are interested in morphology and organisms and you're very good at math or computer science or physics, look into this as a field for you, because there's a lot of science, really good science out there right now that's being done in looking and applying morphology, both on a dissection level all the way into a computerized level. We also see a lot of 3D imagery, so being able to give detailed morphological databases, so that we're still collecting a lot of morphological characters and applying those into our phylogenetic trees. However, there's still really a low number of morphological characters being applied. Some, if you like morphology and 3D imagery, there are free open online libraries called Digimorph or Morphosource, and you can put in your favorite animal and most likely or fossil. You'll be able to have these beautiful 3D images you can play with. We use a lot of these in our studies to look at the morphology and look at that angle. So again, does morphology have a place in today's evolutionary biology? It helps establish species and clades, so it does help that. It was the original way of running these phylogenetic trees. It allows extinct taxa to be integrated into trees of living taxa, so that's where I was saying the fossils. It also helps reveal a suite of phenotypic novelties that characterize molecular groups. So it can be those phenotypes are related to the genotype. And it helps identify convergence that has possibly occurred by doing that character mapping. So my take home for this is, yeah, it does. Morphology does still have a place in our evolutionary studies. Okay, so with that, I'm going to wrap it up. I'd like to thank CASE, which has brought this to you for the last couple of years, and my fellow Darwin Day committee members, Dr. Caitlin Fisher-Reed and Dr. Ellen Igmason. And then, of course, I'd love to thank you guys for participating in this, and I hope you learned something. If not, I hope you learn to appreciate evolutionary biology and appreciate bats. So thank you so much for participating in this.